Welcome to our webinar titled Molecular Genetics of MDS MPN Overlap. Thank you for joining us. My name is Lee Clark, Director of Patient Services, and I'll be moderating the presentation today. As we get started, I'd like to recognize the generous support of our corporate sponsors and the generous support of our patients, families, and caregivers for supporting the webinar today. Due to the high volume of teleconferences on the internet, it is possible you may lose your connection during the program. If you're unable to view the webinar online, you can call in to hear the audio portion of the program using the call-in number in your reminder email. Today's program will be archived to our website within 2 to 3 business days. You will be notified by email when it is live and ready for viewing. Immediately following the presentation, there will be a question and answer session. Please feel free to submit your questions at any time during the presentation. To submit a question or comment, open the Q&A window in the lower part of your screen. Type the question in the small text box window, and when you have finished typing, just hit enter. We'll do our best to get to everyone's questions today. When asking questions, I do ask that you do 2 things to help me manage the incoming questions. First, submit your entire question all at the same time. Second, please do not share private health information in your question. Our speaker cannot answer any specific questions related to your personal health care. Today's specialist is Anthony Hunter, who is Dr. Hunter is an associate professor in the Department of Hematology and Medical Oncology at Emory University School of Medicine. Dr. Hunter specializes in treating patients with leukemia and other myeloid malignancies, including MPNs, MDS, and the MDS-MPN overlap. He holds professional memberships with the American Society of Clinical Oncology, American Society of Hematology, and Society of Hematologic Oncology. Dr. Hunter received his medical degree from the Medical Oncology of Wisconsin in Milwaukee. He completed his residency in internal medicine at Emory University School of Medicine in Atlanta, Georgia. He completed a fellowship in hematology medical oncology at the H. Lee Moffitt Cancer Center and Research Institute in Tampa, Florida, where he served as the chief fellow in his final year. With that said, it is my pleasure to welcome Dr. Hunter today.
All right, hopefully you can all see my screen. Thank you very much for that kind introduction, Lee. It's a pleasure to have the opportunity to speak to you guys today on the molecular genetics of MDS MPN overlap syndromes. I have no relevant conflicts of interest for today's talk, but I will discuss some off-label use of therapies in this presentation. So here's our objectives for the presentation today. We're going to review the MDS MPN overlap syndromes and talk about some of the clinical features and diagnostic criteria for these diseases. And then we're going to highlight common genetic changes in these diseases and the role that this plays in diagnosis, discuss how this impacts determining prognosis, as well as how we use this in informing our treatment decision-making. So some background and historical context in these diseases. The myelodysplastic myeloproliferative neoplasms, and these are also kind of abbreviated as MDS-MPNs, you might hear me say, or just overlap syndromes. But these are a relatively rare group of bone marrow cancers, and we refer to this kind of group of bone marrow cancers as myeloid malignancies. And historically, these have kind of had a serendipitous evolution in what we've named them and how we've diagnosed them. And it wasn't actually until 2001 that these were really categorized as their own distinct category within our kind of classification system of these sorts of diseases by the World Health Organization. I mean, even since that time, new diseases have been added to this group. Really before this, they were just categorized as other related diseases like myelodysplastic syndromes, and really not thought of as necessarily their own diseases until more recently. And largely due to this, as well as the relative rarity of these diseases, they've historically been relatively poorly understood and have had a limited number of treatment options. Now, what makes these overlap syndromes? And so again, these are in a family of what we call myeloid malignancies or bone marrow cancers. I have here kind of and these diseases all exist on a spectrum. And here on the bottom, we have some of the chronic types of bone marrow cancers we deal with. On one end, we have myelodysplastic syndrome, or MDS. MDS is characterized by mostly what we call dysplastic features, and predominantly this is going to be low blood counts and complications related to that, and requiring red blood cell transfusions or platelet transfusions, often a lot of fatigue related to anemia, sometimes bleeding related to low platelet counts or infections related to low white blood cells. And then when we look in the bone marrow, what we see is what we call dysplasia, which is usually a way to say that we see abnormal looking cells under the microscope, and really the inability of the bone marrow to effectively make new blood cells for us. On the other end, we have myeloproliferative neoplasms, or MPNs. And these kind of do the opposite. We see what we call proliferative features, which typically are defined by elevated blood counts or increased blood counts. Oftentimes we'll see things like an enlarged spleen, occasionally a large liver, and a number of other what we call constitutional or kind of general symptoms that can go along with these, like fevers, night sweats, weight loss, bone pains, and occasionally things like skin rashes from the cancer cells themselves getting into the skin. And really in the bone marrow, we more so see just a lot of increase in cells and increased production of these blood cells. Now, these MDS MPNs, or overlap syndromes, really do fit in between these kind of two classes of diseases. And in some cases, they can behave more like an MDS and be on the left side of the spectrum here. And in some cases, they can behave more like an MPN. In some cases, they really have kind of overlapping features, some blood counts that are low, some blood counts that are high. Or an individual patient may kind of move along this spectrum. They may have low blood counts at one point, but as the disease changes over time, may see more elevated blood counts. So really these kind of overlapping features of two kind of distinct groups of diseases is how these were kind of termed and how these kind of came to be as overlap syndromes. So there's now 5 total diseases within this category that are recognized. I have JMML, or juvenile myelomonocytic leukemia, on the right here. That's actually a pediatric disease. And then in the 4 predominantly adult diseases we see, the first is chronic myelomonocytic leukemia. We abbreviate that as CMML. Atypical chronic myeloid leukemia as ACML. And the longest name here is myelodysplastic myeloproliferative neoplasm with ring sideroblasts and thrombocytosis, or MDS MPN-RS-T. And then the last is MDS MPN unclassifiable, which, as we'll talk about it, is kind of a bucket for all kind of rest of patients that don't necessarily meet criteria for these other distinct entities. So before we go on and kind of talk in depth about the genetic changes that we see in these diseases, I did kind of want to highlight a little bit just to make sure we're all on the same page and kind of understanding what these DNA changes are and what kind of tests you're going to see us using to identify these. So this is a kind of a representation of a cell in our body, and essentially anyway, all the cells in our body have a full complement of our DNA. And that's divided up into 46 total chromosomes, or really 23 pairs of identical chromosomes. And these chromosomes are really larger structures of DNA, and that really can contain anywhere from hundreds to sometimes thousands of genes on each chromosome. But as we move down here, we get into kind of the smallest or molecular aspect of DNA. This is what we call the double helix structure that you may have seen kind of pictures of and heard about before. At our very most basic level, we have what are called base pairs. And we have 4 different ones, and these are represented by the letters C and G, which pair together, and then A and T, which pair together. And these letters are kind of analogous to like the zeros and ones you think about with like computer coding, that really these are code that's ultimately used by our cells to make what are called proteins. And then proteins are what ultimately go on to carry out functions or kind of make the structure of our cells, all the kind of building blocks and things that we need. So when we're talking about DNA changes, that can kind of be on 2 different levels. On one side, we can see these chromosomes here with additional chromosomes, missing pieces, and obviously that's going to affect potentially many, many genes. On the other hand, and what we're largely going to talk about today are what we call DNA mutations or changes in the DNA. And these are largely at the level of these base pairs or these individual letters here, and where one letter may be changed to another, and a small number of them may be cut out, and we're kind of small numbers or kind of small changes at that level. And what we kind of understand now, and we've learned more and more about, is that really these mutations in our bone marrow cells, in particular stem cells in the bone marrow, seem to occur all the time in all of us, and really are a function of something that happens as we age. So this table is a little bit complicated, but with each column here that I kind of highlight, you can think of that as kind of all your bone marrow cells at a given point in time. And then as we age and kind of as time moves forward, you move to the right here. So we're all born with kind of a full complement of our bone marrow stem cells in most cases. And as we age, as our cells divide, there's chances for mistakes to happen when we make new DNA. Actually, what mutations are these changes in the DNA. And many of these that we're all developing really don't do anything. They don't impact our cells or how they function at all. And so they just kind of live with that or move along with us for the rest of our life. But over time, we can ultimately develop these mutations in specific genes that matter or proteins that matter and impact how our bone marrow cells function. And we kind of term this now as an entity called CHIP, or clonal hematopoiesis of indeterminate potential. And this is really an entity that's been discovered in the last 6 or 7 years or so, where really a not insignificant number of patients are walking around with mutations and kind of leukemia-associated genes like the ones here. But really, if you look at their bone marrow or their blood counts, everything looks completely normal. And only a small fraction of patients are actually going to ultimately go on to develop something like an MDS-MPN syndrome. But these mutations do allow cells to over time kind of expand within your bone marrow and start to make up more and more of your bone marrow. And then over time, as these cells are dividing more quickly, they can develop additional mutations or DNA changes again. And these kind of sequence of mutations can ultimately lead to the development of diseases like MDS or MPNs. As those diseases then change over time, more mutations can happen, which can lead to things like diseases progressing to acute leukemia or becoming resistant to treatments. But this is kind of under our understanding of kind of how these mutations can arise and change over time in these diseases. Now, some of the tests that you kind of hear us talk about in the clinic, some of these are going to look more so at kind of the full chromosomes or those bigger parts of our DNA. And the classic way to do this is called cytogenetics, or we get what's called a karyotype. And this is really just looking under the microscope at kind of bone marrow cells that we were able to grow, and really looking at the structure, counting the chromosomes that we see. So we're going to see, chromosomes that are missing, chromosomes that are extra, big pieces that are missing or kind of swapped between chromosomes. And when we see these kind of abnormalities in around a third of patients with MDS MPN overlap syndromes, these mostly are not diagnostic, and more so can specific chromosome changes really define related diseases. For example, something called the Philadelphia chromosome is exclusively seen essentially in a disease called chronic myeloid leukemia, or really a type of MPN. So really, these changes can kind of help rule out other specific diseases, and in some cases, they do have prognostic information as well. We also have a test called FISH, or fluorescent in situ hybridization. This is, again, looking at chromosomes, but only specific ones that we want to look at. So we design specific probes to look at a specific piece of a chromosome that may be missing or swapped. So we're only going to find exactly what we're looking for. And then the last is what's called a SNP array. And we don't use these quite as much anymore now that we have some of the tests I'll talk about next, but you can detect kind of smaller pieces of chromosomes that are changed or missing that we may not see. Or polymerase chain reaction. This is a way to, again, kind of look at specific things that we know we want to look for. A common one would be this mutation in a gene called JAK2, specifically at this position called 617 in that gene. And that's a common mutation we see in MPN diseases. And sometimes we'll send a PCR just to see if that's there. But really only going to find exactly what you're looking for again here. On the contrary, we have what we call next generation sequencing, or NGS or NGS panels. And that's really what we're going to focus on here, and what we're doing a lot of in the clinic now. And now this test is going to sequence, as opposed to like a PCR, where you're just looking for one specific thing. You're going to look at whole genes, or at least whole big parts of genes, where you'd find kind of any mistake within that section of the DNA that you're looking at. And then typically, as opposed to just looking at one gene, these are looking at many genes. So at our institution, we have a panel that looks at 75 different genes of interest, and that we know are commonly changed or mutated diseases like MDS and MDS-MPNs and leukemia. And because of the number of genes and kind of looking at the whole genes in these cases, we're able to detect abnormalities on these sort of tests in upwards of 90+% of patients with these types of diseases. And not all necessarily impact kind of decision making, but a number certainly can aid in diagnosis and determining prognosis, as well as kind of determining treatment options. And so really these, I consider this to be a standard test in the workup of newly diagnosed, and occasionally we do this kind of multiple time points down the road in these overlap syndromes. And this is what we've kind of found, is that obviously now we're doing this at a clinical level. At first, we were doing a lot of the sequencing more so at a research level. And this has really revolutionized at least the genetic understanding of how we see these diseases. So the table here on the right is from a paper we published a year or two ago, and really breaking down kind of the most common mutations we see in these diseases. Again, and then kind of breaking down what percentage we see them in each of these 5 entities within this category. And these genes really are broken up into a handful of at least the proteins that they code for, how those work in the body. So some are involved in kind of regulating DNA. Some important ones in these diseases are in what we call cell signaling, or really kind of a communication that gets sent through your cells and from other cells to each other to kind of tell them to grow and divide, for example. And you can see really there's a number, about 25 or 30 of kind of more commonly seen genes that we can see across these diseases, as well as other diseases like MDS or acute leukemia like AML, but that are seen at kind of unique frequencies within each disease compared to another. And then in the figure on the left here, this is from one of the most recent papers looking at the genetics of these diseases. I have it listed down here by Dr. Palomo. And this was probably the best and largest paper looking at this so far. And you can see kind of the frequencies of, again, they identified about 30 genes that were more commonly seen as change in these diseases, and kind of the breakdown of the different frequencies that it's seen within each disease. And so we can certainly use this information, and we'll kind of initially talk about how it affects how we make diagnosis and how we determine prognosis in patients. And so classically, how we diagnose patients, well, mostly based on what blood counts look like and what things look like under the bone marrow, under the microscope when we do a bone marrow biopsy. But this is challenging in that these themselves are overlap syndromes, so there's kind of overlap between these diseases. There's overlap with MDS, with MPNs. And so although we have kind of definitive or specific criteria for diagnosing diseases, in individual patients, it's not always so black and white, and there can be a lot more gray area there. And so these mutations can help us kind of say potentially which disease is there, or in some cases, if we don't see a lot of changes in the bone marrow, seeing a DNA change or mutation like a lot of what's called risk stratification and trying to determine, kind of specific groups, higher risk or lower risk, for example, as far as how we think the disease might behave. This again uses things like bone marrow and features and blood counts, and historically more the kind of chromosome changes that we've been able to do for longer. But again, even when we've done this in the past, there's still a lot of variation that we can't account for. And we now have seen that a number of these mutations, although not all of them, do independently predict, how the disease behaves. So I'm going to go through now kind of individually each of these kind of 5 diseases within this category and talk a little bit about the genetics and diagnosis and prognosis. So we'll start with chronic myelomonocytic leukemia, or CMML. And this is the most common of these diseases, and probably the best understood for that reason, although in the grand scheme of things is still a relatively uncommon disease. For each of these diseases on this slide, I'll have the diagnostic criteria here on the right. This is from our World Health Organization, which is an internationally established criteria. You can see the hallmark of CMML is an increase in these type of white blood cells called monocytes. And normally, these are present around 4 to 5% of our blood, but in cases of CMML, we see it making up over 10% of our white blood cells. And then importantly, can't meet criteria for other related diseases. You'll see this criteria for what we call blast cells at less than 20%. Blast cells are kind of the most immature cells in our bone marrow and really represent more leukemia type cells. And that 20% cutoff, anything above that really defines acute myeloid leukemia, or AML. And so within this CML, it really is truly, it kind of fits well into this category. And for that reason, it's kind of been split up into 2 subtypes. And we some patients that behave with a much more proliferative or MPN-like disease. And we use this in kind of our FAB system, define this on a white blood cell count that's elevated, versus patients that behave more like an MDS or a dysplastic type of CML and this is really defined in patients of a lower white blood cell count. And again, this can behave almost exclusively like an MDS, almost exclusively like an MPN. And it's not uncommon to see, for example, a patient who behaves more like an MDS over time to become more proliferative. And then it's also broken up by our kind of World Health Organization into a couple of subgroups. Again, this is just based on those blast cells, kind of leukemia cells, both in the blood and in the bone marrow, and the percentage of cells they're making up. And they divide that into a CMML 0, 1, and 2 in the most recent system anyway. And this does carry some prognostic importance as well. And now getting into the genetics of CMML, for each of these diseases, I'll have kind of a blue box here of kind of the most common or characteristic mutations that represent the disease, as well as the rough frequencies that we kind of see them. And And then I put asterisk next to kind of the most important ones, again, from that kind of paper I mentioned a couple slides ago. That's really the most recent clinical study to look at these genetics. And I'm asterisking ones from that paper that were really the most specific for this particular disease. So TET2, a gene called TET2 is by far the most common in CMML. That's been shown in a number of studies now. It's seen in 60+% of patients in those studies, and it really thought to play a significant role in the kind of biology of the disease. Again, we've talked about kind of multiple mutations developing over time, but it's thought this is one of the first. All being seen in roughly 50% or more of patients. And this kind of unique molecular profile can really help us determine that this is CML compared to another disease. For example, there can be a lot of overlap with MDS and patients who have lower blood counts, and that monocyte count, the number of cells, may be really borderline, kind of right around or even a little bit below the kind of criteria to diagnose CMML. But when we see, for example, this kind of constellation of mutations, it really supports that, this is CMML and not something like MDS. And also a number of CMML patients, we don't see a lot of we call dysplasia or abnormal looking cells in the bone marrow, and seeing these mutations can really help support that increase in monocytes that say it really is due to a cancer of the bone marrow like CMML and not due to an infection or something else going on. You'll see in these studies that a lot of these same genes are popping up in a lot of the different diseases, and although there's kind of characteristic ones, sometimes it's more helpful to use basically a combination of genes. So for example, in CMML, what's found to be really specific for the disease is having at the same time both a TET2 and an SRSFT mutation. And patients that have this kind of combination of mutations are much more likely that this is CMML compared to some other related diseases. And then these signaling mutations, again, these are ones that kind of tell cells to grow and divide faster and not unexpectedly are associated with a more proliferative version of the disease. Now, how does this relate into kind of determining prognosis, or what we call risk stratification? And this is something that there's been a lot of work in over the years in CMML and has been somewhat challenging. And there's actually been about 10 different of these kind of scoring or systems that have been used over time. As you can see, I have them listed, some of the older ones anyway, listed on the bottom here. 4 of these weren't even unique to CMML patients and really were more made for MDS. 3 of them were kind of specific to CMML, but didn't incorporate things like genetic data. And so really, which is the best has been hard to determine. A mentor of mine, Dr. Padron down at Moffitt, had published a study a few years back that looked at these 7 here that are listed, and really found that although they all do help predict prognosis, none is necessarily all that good, and none was necessarily better than the other. And then also showed that in this paper and many others that in addition to the kind of usual things we use to look at prognosis, these mutations very much do impact prognosis as well. And so we've now developed, not me specifically, but the community has developed 3 specific models of these kind of systems to help look at prognosis in CMML patients, all of which include genetic data. And I have those 3 listed here. The first is called the GFM model, which includes ASXL1 mutations. Mayo molecular model also includes ASXL1, and then CPSS molecular model, which includes a handful of mutations. And so what you can see is that all of these do include what's called ASXL1 gene. And really kind of across all genetic studies of CMML, this is kind of the one gene that has always stood out as most conclusively to impact prognosis, and in particular with a worse prognosis or a shortened survival with this disease. And again, that's now incorporated in all 3 of these models and really has been shown kind of time and time again. A number of others that I have listed here have been shown, and for example, the CPSS mole model has a couple different mutations, and there have been some others in individual studies, but maybe not quite as definitively as ASXL1, which can be limitations of individual studies and things as well. But a number here that have had at least some suggestion of also correlating with kind of worse outcomes. And then TET2, which we mentioned is the most common gene in the disease. This is not included, as you can see, in these models, and our kind of our understanding that gene has changed over time. But I was recently part of a study that was led by the group at Mayo, who does a lot of work in these diseases. And we combined databases from 3 major cancer centers to look at over 1,000 patients, and found that really these TET2 mutations were associated with a more favorable impact of the disease. And this was particularly true without ASXL1 mutations, or in patients who did have that more negative mutation. They almost kind of balanced each other out, and to be at least kind of more towards a kind of neutral between the 2. And then kind of lastly is these RAS mutations. This is again a type of signaling mutation and can be associated with the disease kind of progressing or changing over time. But the next disease we'll talk about here is JMML. Again, this is a pediatric disease, so it's actually not one that I treat at all, but and that's because it's really diagnosed in infancy and predominantly in children less than 3 years of age and in over 3/4 of cases. Again, it's kind of analogous in a number of ways to CMML. It's kind of defined by this increase in the monocytes and this particular type of white blood cells. These patients almost all will have an enlarged spleen. Often will have other symptoms like fevers, rashes, enlarged lymph nodes, and a number of other kind of systemic symptoms going on. And you can see from the diagnostic criteria here that, in this disease, these mutations really have made their way truly as kind of a requirement in the diagnosis of this disease. And then an important note is that this JMML can actually arise in the setting of congenital syndromes. So Noonan syndrome and neurofibromatosis type 1 being the most common. And now this disease is considered what we call a RASopathy. So RAS, I think you've heard me mention a couple of times. This fits into, it's really a couple different RAS genes, but they fit into what we call a signaling pathway. So signaling pathways are kind of like a game of telephone where our cells, kind of one protein gets turned on, that turns another one on, turns another one on, and ultimately that kind of carries out some sort of change in a cell, like telling it to grow or divide more quickly. And these RAS pathway, there's a number of genes involved, and all of the ones listed here under the characteristic mutations are all within this RAS family of genes. So you see 2 RAS mutations, NMK-RAS, CBL and NF1, for example, turn the RAS pathway off. And so what you see is that over 90% of patients with this disease have one of these RAS pathway mutations, and these are the ones that have now been included in that diagnostic criteria. Again, these mutations can be what we call a germline or inherited, or ones you're born with, or these can be ones that develop, which we call acquired or somatic mutations is the terminology for those. And we do see some clinical variation based on, kind of which mutation is also whether it's germline or somatic, as we call it. Now, some of the other mutations that we've shown on some of the other slides, and other genes, not so much as common in this disease as others. And we do see them to some degree at kind of smaller levels. In one study, SEPBP1 and JAK3 were seen in around 15% of patients. And some of these other mutations can be seen, but much more rarely than what we see in some of our other adult diseases. But certainly these do impact prognosis. So in particular in the setting of some of our inherited syndromes. So Noonan syndrome, which mostly is caused by mutations, or at least half of the time by mutations in PTPN11. These patients or children can develop what we call a transient myeloproliferative disorder. So it's really an MPN-like disease that actually resolves over time, often without treatment. And in some forms, this can very much resemble JMML. In patients where the disease is driven by this CBL gene, again, this is one that's usually inherited. And again, this is one that can resolve on its own over time. And then that's occasionally seen very rarely with NRAS mutations as well. And then we see these double RAS mutations, or kind of 2 mutations in RAS pathway genes. And these were felt to not really necessarily happen 2 at a time, but now we do see that they truly can occur and tend to behave more aggressively when we see that. And then lastly, these kind of other mutations that aren't in the RAS pathway, the ones that we mentioned in disease like CMML, or although uncommon, do suggest kind of more aggressive or the disease at a later standpoint and the disease progressing over time. Kind of back to the adult syndromes, we'll talk about atypical CML. This got its name because really from what the blood counts look like predominantly is very much like what we see in a disease called chronic myeloid leukemia, or CML. But CML is defined by this abnormal chromosome, or the Philadelphia chromosome, part of the 9th and 22nd chromosomes getting stuck together basically. And that's negative and must be so, for example, to diagnose this disease. So predominantly what we're seeing in these patients are a high white blood cell count, and then what we call dysgranulopolysis or dysplasia, just meaning that these white blood cells look very abnormal. They're developing abnormally. And then we often will see anemia, other low platelet counts, and large spleens and things as well. And this does tend to, at least on average, be the most aggressive of these MDS MPN overlaps. Now, this subsequent diseases aren't understood quite as well as CMML because they are a little bit more rare and somewhat harder to diagnose in the past. And so there's been some variability among studies, but ASXL1 does seem to be the gene that's been kind of most correlated with this disease. And kind of older studies, anywhere in the kind of 40 to 60% range of patients will have had this. But in the most recent study by Palomo et al. That I mentioned a couple of times now, over 90% of patients actually had this gene mutation. In addition, some other mutations that are common, again, SRSF2, SEPBP1 genes that we've seen in a disease like CMML. But SETBP1 and then this other kind of more rare mutation in a gene called ETNK1 are seem to be more specific for the disease. So if we see, kind of features of an overlap syndrome, see this, for example, ETNK1 mutation, very much suggestive of this disease. And just like with CMML, certain combinations also very much more and kind of favor this diagnosis, and really combinations using any of the three genes, and particularly ASXL1 and SETBP1 combined are really suggestive of this diagnosis. And again, when clinical features can be really kind of hard to determine, sometimes seeing this combination can really favor that this is the disease that we're dealing with. Some older studies suggest that this gene called CSF3R was common, but really, although it's present in probably around 5 to 10% of patients, it's mostly specific to a type of MPN called chronic neutrophilic leukemia, where it's seen in the vast majority of patients. And how these genes impact prognosis of atypical CML is not quite as well defined. And again, in this most recent study I've mentioned a few times, a couple of genes here, SRSF2 and SETBP1, were felt to be more favorable, and whereas several others were associated with a worse prognosis. And in that study, although ASXL1 in the whole group of patients was an inferior or kind of a mutation that predicted worse prognosis, it didn't seem that way in atypical CML, with the caveat that over 90% of patients did have this mutation in atypical CML, and this disease did have the worst prognosis compared to other diseases, so still certainly seems to be playing kind of a role in prognosis there. One study by the Mayo group identified TET2 as kind of associated with the worst outcomes. So they did develop kind of the only prognostic score in this disease currently, and looking at this mutation as well as age and being anemic with a hemoglobin of less than 10. And then SETBP1, which again in this most recent study was a more favorable mutation to have. Some of the initial studies showed it was actually a worse mutation to have. It was associated with a worse survival, and several in between there, it was kind of neutral. So I'd say the data is still a little unclear on that particular gene. Next disease is MDS MPN-RS-T, and I really think of this as kind of the quintessential overlap syndrome. It's truly like combining both the clinical features and the genetics of two diseases. And those diseases are MDS with ring sideroblasts. This is a type of anemia, and that predominantly causes, type of MDS that predominantly causes anemia. And you can see anemia is one of the diagnostic criteria here. And then it combines that with a disease called essential thrombocytosis, or one of our most common MPNs, which typically causes an increase in platelets. And you can see that's also part of the diagnostic criteria. So these patients really do have typically anemia and a high platelet count. And then as part of the name and what I have pictured here on the bottom right, these are a type of cell called ring sideroblasts, which really are just a type of cell we see under the microscope in the bone marrow. And when we stain for iron, and these little blue dots kind of form a circle around the middle part of the cell, and that's kind of abnormal iron accumulation. That's kind of characteristic of this and the related MDS disease. This disease typically has the best prognosis of these MDS MPNs, but there is some increased risk of blood clot, as is also seen with ET. As far as the genetics of the disease, it seems to be the least heterogeneous of, at least of the adult MPNs, and kind of the lowest average number of mutations. It's really driven for the most part by a gene called SF3B1. And this is seen in upwards of 85+% of patients. It's thought to be kind of the founder or initial mutation for the disease. It's what accounts for those ring sideroblast cells, and also is seen in a similar frequency in the MDS with ring sideroblasts that this disease kind of comes from. And then the second gene we see most commonly is called JAK2. And this is seen in at least half of patients. And this is the most common mutation in the MPN-ET that causes high platelet counts. So that is what kind of gives that aspect of the disease, or the MPN component, is this JAK2 mutation. And we do see some other mutations, here as well. And again, this kind of combination of these two genes, SF3B1 and JAK2, is highly suggestive of the disease. And I've diagnosed several patients with this disease based on this genetic findings when the bone marrow was a little bit unclear. And as far as prognosis, this is very much a disease where any chromosome, kind of full pieces of the chromosome change, has been shown to be associated with worse outcomes. And then a number of mutations, ASXL1, which shows up a lot in this, and a couple others have been associated in at least some papers as having a kind of worse survival for patients. And again, the Mayo group here did develop a prognostic score that included 2 of these mutations, as well as having any abnormal chromosome change, and then being anemic, which is a common component of a lot of our scoring systems. Some earlier studies suggested that SF3B1 and JAK2 mutations were better to have. But this hasn't been shown in other studies, and really these probably more so just define this disease, which itself does have a more favorable prognosis compared to some of the other MDS-MPNs. And so the last one we'll talk about here is MDS-MPN-U, or unclassifiable. This is basically kind of a basket diagnosis for patients who have kind of both dysplastic or MDS-like features combined with MPN-like features. And that's how it kind of gets in what the diagnostic criteria are made of kind of looking like an MDS, but also having a high white cell count or a high platelet count, sort of like an MPN. And so really patients who just don't fit any of the other criteria for these diseases, but have these features. And for that reason, it's a fairly heterogeneous group of patients. For that reason, it's one of the least genetically characterized of the group. Again, you'll see a lot of these kind of same mutations popping up as kind of the characteristic ones. The ones that were kind of found in the recent study I mentioned to be most specific were TP53 and U2AF1, which interestingly, neither were all that common in the disease, but for example, TP53 is a very important mutation due to its prognostic implications, and it's quite rare in other diseases like CMML, although it is more common in diseases like MDS. But in this study, they were actually able to kind of break these patients up into a couple different groups, largely trying to make them more like one of these other specific diseases within the group. I mean, they did that solely based on the genetics and particular combinations of genes that patients had. So about a third of patients genetically were like an atypical CML, about 17% like a CMML, and 11% like an MDS MPN-RS-T, for example, having the SF3B1 and JAK2 combination that we talked about. And then a fourth group of patients that have the TP53 mutation, which again is unique because it's not typically seen in these other diseases, and about a quarter of patients that really didn't fit into any of these categories. What they found is this kind of breaking patients down into kind of the disease that they're most similar to from a genetic standpoint was the biggest kind of impact on predicting prognosis. So we saw that, before we've discussed that MDS MPN-RS-T is kind of the most favorable disease to have. And so the patients who have genetics that look a lot like that disease did the best. Again, atypical CML was of these diseases kind of the more aggressive one typically, and that kind of also, was similar here. And then lastly, these TP53 mutated patients, again, this is a gene that kind of across bone marrow cancers tends to be correlated with a kind of worse outcomes and increased progression to leukemia and worse survival. And that's very much what they saw with this disease as well. There are some individual genes that have been associated with, kind of an inferior or kind of worse prognosis. Again, TP53 being the most common or most conclusive one here. One study did identify this CALR, or kind of an MPN-like mutation, as being better one to have, but it was only seen in a couple % of patients, so really is not all that particularly significant from a clinical standpoint. So moving on to treatment for the rest of the talk here. It's just kind of an overview. Treatment options, as I kind of alluded to before, have historically been somewhat limited in this disease, or these diseases, and that's partly due to how rare they are. Partly due to, again, some of these diseases didn't even have names until the last 10 to 15 years. And so it's been harder to, for example, do specific clinical trials to look at these diseases by themselves. And really, occasionally a small number of these patients got into trials, for example, of MDS. And really that's how treatment has historically been done, is we just kind of treat patients like how we treat MPNs or how we treat MDS, at least historically. Some of the kind of decision-making or factors that go into that decision-making I have here, and probably number one is how are patients presenting? What are their symptoms? What do their blood counts look like? Is this more of an MDS-like disease where we're dealing with low blood counts that we need to improve? It's more of an MPN-like disease where we're dealing with things like a big spleen that we need to treat. And really we have kind of different treatments for the most part to target those different things. Another important aspect is being able to, at least best we can, predict the aggressiveness of the disease and therefore how aggressive we need to be. One of the things obviously that are important, not all patients can tolerate, for example, very aggressive things like a bone marrow transplant, where some patients don't want to go through with those sorts of things. So I always, kind of discuss with my patients that this is really a team approach, that we need to kind of work together to come up with the best treatment on an individual patient basis. And I do highlight clinical trials. I think if clinical trials are available, and that's always a good idea given our kind of, overall limitations in these diseases, and should definitely be considered. And then I will point out here that bone marrow transplant is the only thing that, and I will highlight potentially curative, because it's potential but not a guarantee, but the only potentially curative therapy in these diseases. So here's a kind of treatment algorithm that this was from the paper I published in CMML a couple of years ago. So this is clinically unique to CMML, but really does kind of apply for the most part to all of these diseases. So again, the kind of initial branch point and how we're determining treatment is determining how aggressive we think the disease is going to be, using the genetics, using some of the scoring systems to kind of help us determine that. In low-risk patients, now we're kind of getting into how patients are presenting. Some low-risk patients don't have symptoms, their blood counts aren't all that bad and can be watched sometimes for years and without needing treatment. And some patients may have predominantly anemia or low hemoglobin, and we can treat those with things what we call erythropoiesis stimulating agents or EPO drugs like Aranesp or Procrit, and just kind of targeting the anemia. And then I have an asterisk here because specifically in MDS, MPN-RS-T, there's a couple other treatment options. Lenalidomide is one, but in particular, luspatercept, which is a newer drug that's been approved within the last couple of years specifically for this and the related MDS disease. And then some patients have more proliferative features, things like a big spleen or elevated blood counts. Hydroxyurea or hydria has been kind of the standard here for a long time. And we have published recently a phase 1 and 2 trial looking at ruxolitinib and CMML patients and did some activity there. And so certainly that's an option in a number of these diseases as well. And patients who have more predicted anyway to be more aggressive disease, this is where we really want to consider a bone marrow transplant in anyone who's a potential candidate and who has a donor. And when we can't, or when we can at least go to a transplant right away, again, we're kind of getting back to how patients are presenting. So if we're dealing mostly with low blood counts, we use a class of drugs called hypomethylating agents or HMAs. There's two drugs here called azacitidine and decitabine, and these are kind of our standard treatments for myelodysplastic syndrome or MDS, and really geared to improve blood counts over time, hopefully help decrease things like those blasts or leukemia cells, prevent progression over time. They've been shown to improve survival in MDS specifically. And proliferative features, kind of the same options as before, and then in patients who have kind of combined these or high risk for having these high levels of blast cells, again, these hypomethylating agent drugs, azacitidine and decitabine are often kind of our standard, although may need to be combined with some other things to help control the disease. We'll kind of highlight now, how we can use this genetic information to help us. And there's kind of three places that I think it's more important to try and use this data. And one is, again, looking at and trying to guess or determine predict, I should say, the aggressiveness of these diseases. And really that helps us know how aggressive and urgent we need to be with our treatment. Number two is can we predict which therapy, if we have multiple options, or if we don't have many options, if a standard treatment is likely to work? And then lastly is, do we have treatments that target specific DNA changes within an individual patient? So look at the first of these is identifying kind of appropriate treatment strategies by best predicting how we think the disease will behave. Again, we've talked about this with kind of each of the diseases, and again, classically use things like blood counts and bone marrow findings, with the genetic data really, especially in diseases like CMML, really adding to that and our ability to do that. And this is important in a couple options, and I think in particular kind of at each end of the spectrum. So on one side, it's very important for us to know and predict patients who are truly going to be low risk. And these patients, again, unlikely for the disease to progress over the time, and oftentimes don't have a lot of symptoms. And so these are patients we can treat, or sorry, not necessarily treat for some time. And we've had patients that we've observed for years or even a decade more. And so identifying the patients who we think really are likely to do well off treatment, is important. Whereas, some patients who classically may have looked low risk based on their blood counts, we're now learning from a genetic standpoint may not be so low risk and therefore much more likely to progress more quickly. And then second is on the other end is, who really are the higher risk patients that we really need to be thinking about bone marrow transplants for. Again, I counsel my patients that, bone marrow transplant is a high risk, high reward strategy, right? It's the only treatment that we have with the potential to cure patients, but that does not come without risks. And that risk can include patients dying, much quicker than they would have if we hadn't done a transplant. So it's important that, we're taking the right patients to transplant, both from a disease standpoint and from a patient standpoint, about who's likely to tolerate it, who's likely to get the most benefit out of it. As far as kind of option number two, which is using this data to predict our, kind of how well our usual treatment's going to work. And we've made some improvements here, although still some work to do, certainly. Again, these hypomethylating agents, azacitidine and decitabine, are our most common treatments. And we've seen this in a number of kind of these bone marrow cancers, certainly in MDS and CMML. TET2 mutations, which again are the most common in CMML, seem to associate with a better chance of responding to these drugs, a better chance of blood counts improving. And this is particularly true in patients who don't have an ASXL1 mutation. Again, they kind of balance each other out to some degree, and ASXL1 has been suggested to have worse response rates to these drugs. And then as far as kind of independent of response is kind of how survival and how patients do long-term with these drugs. And again, at least uniquely to CMML, TET2 seems to be more favorable, whereas some of these other mutations like TP53 and RUNX1 appear to be worse. And I mentioned the drug luspatercept a couple of slides ago, and this is a treatment that's really geared towards just improving the red blood cell count or improving anemia. And this was studied and is approved now in MDS, MPN-RS-T, and more so it was studied in the kind of related MDS syndrome or MDS with ring sideroblasts. Again, these two diseases share this mutation in SF3B1 in most cases, and in early studies of this drug, these patients with these mutations and with those ring sideroblast cells that we saw under the microscope, those were the most likely to benefit. And so that's where this drug has been approved now and is a standard treatment available to anybody, kind of meeting the criteria as far as anemia and things like that. And then lastly, bone marrow transplants, again, is one of our, theoretically standard approaches. And can we, although we are using mutations to help predict who we should think about transplant because the disease can behave aggressively, we can also potentially use, the mutations and other data to predict, who's likely to do well with transplant, to stay cured long-term. And for example, in CMML, we've seen that this ASXL1, which again, we mentioned pops up all the time in this disease, as well as NRAS mutations, were suggested to be kind of worse overall long-term outcomes for patients going to transplant with CMML. And then lastly, is what we call molecularly targeted therapies. So these are drugs that, typically pills that are geared to treat specific or target specific mutations, meaning they work if you have that mutation and they don't if you don't have it. And really along with immunotherapy or using the immune system has really revolutionized how we treat, cancer and the entire field of oncology now. But none of these drugs are currently approved in these MDS MPN syndromes. Some that we do use or are looking at using, of course, ruxolitinib, I mentioned before in the CMML algorithm. And this is we call it JAK2 inhibitor. So it kind of targets that JAK2 gene and that pathway, which can be activated in this disease. And we did see in a phase 1/2 study, again, that was just published within the last couple of months, that there was certainly some activity seen in CMML patients, which did not depend on which mutation was there. It's also been studied in combination with azacitidine, one of those HMA drugs we've mentioned, and some of our MDS, MPN-U, and other overlap syndromes. And it did seem in that study that there were some improved responses with JAK2 mutations. And then lastly, these CSF3R mutations that I mentioned are mostly seen in CNL, but they can be seen in a small subset of patients with atypical CML, and then rarely in 5% or so of less of CMML patients, and does suggest a sensitivity to this drug as well. Another mutation that we haven't really talked about so far, there's two genes, one called IDH1, one called IDH2. These are only present in around 5% of patients with these diseases. But important is we have now pills, ivosidenib and enasidenib, that target these genes and are approved now in acute myeloid leukemia, so kind of acute leukemia version that these diseases can progress to. And so although they're not currently approved, they are being investigated in MDS, for example. There's been some studies in more accelerated versions or aggressive versions of MPNs. And so likely that the kind of indications or diseases that we can use, these will expand over time. And certainly they're available for patients who progress to acute leukemia. We occasionally use them off-label as well. And then lastly, there are some other pathways and number really in investigational approaches. A couple that I highlighted here. This drug H3B8800 is targeting these what we call spliceosome genes. There's 4 of them, and I think SF3B1 and SRSF2 are both ones that came up in a number of these slides before. And this drug, and at least in this initial study, didn't show a ton of activity, but is still under investigation. And then this RAS pathway, the signaling pathway that we've talked about, is really common across cancers of all different types. And so there's been a lot of investigation over time and ongoing to look at ways to target that pathway as well. And certainly more that we will continue hopefully to develop as we move forward. And so in conclusion, we've highlighted that these MDS MPN overlap syndromes are a relatively uncommon group of bone marrow cancers and do tend to be quite variable, and for that reason somewhat challenging to diagnose and challenging to treat. And we talked about some of our advancements in this DNA sequencing technology or ways to evaluate DNA changes that are now widely available in the clinic and do improve our understanding of how these diseases arise, how they progress over time, and certainly a number of mutations that we commonly see. And that we can use these now to, at least in some cases, play a role in aiding us in diagnosis and accurately doing that. trying to better predict prognosis and then also determining treatment selection. And so this is something that I do in all of my patients and really should be considered standard of care in anybody with any sort of kind of myeloid malignancy or bone marrow cancer like this at this point. And so at this point, I'm happy to answer any questions and appreciate you guys listening to the talk.
Thank you so much, Dr. Hunter. We have a few questions. Our first question is, next-gen sequencing was not available in 2008. Is it possible to run NGSS on historical bone marrow biopsies to look at mutations at the onset of disease?
It is. So a lot of, and it depends on the panel. There's a lot of these available. So a lot of like bigger cancer centers and institutions like ourselves, we have a panel that we just do in our own lab here, which is nice because we get it back a little quicker and we can see the data and that sort of thing. Now, not all of them can you go back and use kind of historical bone marrow samples. And a lot of them will require kind of more fresh samples, but it just depends on the actual, which type of test you're using. There's a lot of commercially available ones. So some of these, I believe Foundation One, which is kind of a big company that does this, a lot of sequencing in both bone marrow cancers, but also solid tumors. And typically some of theirs you can potentially do on at least adequately kind of preserved kind of whole bone marrow samples from previously. And so that is potentially an option. It can be challenging depending on how far back you go and how well the sample was preserved and things like that, but it is a potential option. And certainly this is something that, I have a lot of patients who come to see me as kind of second opinions and consults and have not had this done. And it's hard sometimes to go back and get old samples, especially if they're from other centers and things like that. But it's still something that, although I like to get it done at diagnosis, it's certainly, if you've never had it done, something that, I recommend doing at any point to kind of help determine where the disease is at that point. And in some patients, I'll do this kind of periodically if, for example, they had really low grade disease at one point and we could watch for a couple of years, and then in 3 or 4 years, the disease is clearly getting more aggressive, we may repeat that sequencing test to look for new mutations that have developed that suggest we're going to, move on to something like a leukemia, or if it's moved on to a leukemia, looking for mutations that we can target with kind of pill treatments, which we have more so approved in leukemia at this point.
Thank you. Our next question is, does having a MDS-MPN overlap complicate the success of a transplant?
So you mean like for bone marrow transplants for MDS-MPN overlap syndromes, I would say, certainly these have been rare disease. We don't have as much data as we do, for example, like acute leukemia and things like that. But certainly we routinely do transplant patients with things like CMML, and again, it's the only kind of long-term cure. Now, certainly the potential outcomes with transplant is going to depend on a lot of factors. Some of that is dependent on things like how healthy a patient is to begin with. We know the healthier a patient is, the better they're going to come out from a transplant. Again, a number of these genetic and kind of factors of the disease can affect how successful a transplant is, and there can be things that we can't predict, obviously. But certainly, kind of an indication for a transplant, and there's no guarantees in the transplant world, but certainly a potential indication to move forward with something like that.
Thank you. Our next question reads, what is known about the KRAS mutation found in CMML1 11 years after onset of disease? So is it likely to have a positive effect on prognosis?
So KRAS and CMML typically not associated with a more favorable prognosis. Those RAS mutations are ones that more so we see that in the more proliferative type of CMML, the kind with that behaves more like an MPN with higher blood counts or enlarged spleen. It tends to be a more proliferative mutation just because of the kind of way that it works in the cells. But again, even genetics, I think this gets to that point, is that we can't explain everything. Even genetics adds a lot to just looking at blood counts and things like we used to do, but there's still things that we can't necessarily predict. There's still plenty of variability. So although KRAS is one that typically I would think of as more one that's associated with a proliferative type of CMML, and some studies has been suggested to be associated with a worse prognosis, although certainly not in all of them, but certainly not necessarily one that's more favorable.
Thank you. This question comes from a CMML patient with AXL1 BAF 13%, TET2 45.3%, and CRSF2 22.9%. They'd like to know, could the TET2 mutation cancel out the bad prognosis of their ASXL mutation?
Yeah, so that is something that we've seen. So again, I mentioned a study that I was part of. It was led by some collaborators at Mayo, where we combined and looked at over 1,000 patients with CMML to get a better idea of truly how do TET2 mutations impact things. And TET2 on its own was clearly more favorable to have, especially it did somewhat depend on which type of TET2 mutation, or for example, having two TET2 mutations is better than having one. But certainly has been associated with kind of more favorable. Again, ASXL1 as kind of more negative, but at least in that study, which is the biggest, looking at this, certainly, they did to some degree balance each other out. And maybe not 100%, but certainly the TET2 mutations definitely had some, potentiator kind of counteracted the negative aspect of that ASXL1 mutation. And we see that also with, kind of responding to drugs like azacitidine, is that, having TET2 is usually good, having ASXL1 is kind of bad, and kind of in between, having both of those is, or neither, is kind of somewhere in the middle in most cases.
Thank you so much, Dr. Hunter, for sharing your time and your expertise with all of us today. If you'd like to watch this webinar again at a later time, it will be available on our website within 2 to 3 business days. If you have any additional questions, please send it to us at any time in 3 ways. Send an email to help@aamds.org, call our helpline at 800-747-2820, extension 2, or submit your question to our Facebook page. On behalf of the Aplastic Anemia and MDS International Foundation, thank you for joining us today, making us your resource of choice for information on bone marrow failure diseases. Our medical advisory board and team are always here to support you and your family, as we've done for the past 37 years. This concludes our program. Thank you so much, Dr. Hunter.
Thank you for having me.
Have a good day.
You as well.